Following endosperm development, there comes another post-fertilization event, that is embryo development. As discussed earlier, the development of embryo starts due to the cellular division of the zygote that is located near the micropylar end. In most of the cases, this embryo development starts only after a certain amount of endosperm is developed because it provides an assurity that there are reserves of food material for the developing embryo. And therefore, embryo development starts only after the development of endosperm tissue occurs. Because it provides an assurity that there are reserves of food material for the developing embryo. Now within an embryo, there occurs mainly two types of cells. These two types of cells generally occur in dicotyledonous plants like Capsula, which is a dicot plant where mainly two types of cells occur during embryo development. First is basal cell. Basal cell is the cell that occurs towards the chalazal end and is larger in structure. And the second type of cell that occurs is apical cell that occurs towards the base, that is micropylar end. And is small in structure. It is because of the division of these cells that the embryo development occurs. As it can be seen from the figure, it is the figure of the embryo development within the dicot plant, that is Capsula. As here it can be seen, the basal cell, which is the larger cell, is formed by the zygotic division, which also results into the formation of terminal, that is apical cell, which is located towards the micropylar end. It is because of division of these cells that entire embryonic structure develops. In this structure, zygotic division forms the apical and basal cell. Out of which the basal cell divides transversely and the apical cell divides vertically. This results into the formation of a four-celled structure, and this four-celled structure is known as proembryo, which is formed by the division of apical and basal cell. This proembryo is the basic of starting of the structure of embryo. This proembryo divides to form first of all the globular structure, which then converts into a heart-shaped structure, and finally mature state, that is torpedo stage. Thus, proembryo divides to form a globular structure, which further divides to form a heart-shaped structure, and finally results into mature form, that is torpedo structure. However, there are a few times when the proper division of the basal cell within embryo does not occur. At that time, the basal cell further divides to form a filamentous structure of about 20 to 25 cells, which is known as suspensor. Thus, if the two basal cells do not divide transversely, then it results into the formation of a filamentous structure which is of about 20 to 25 cells, known as suspensor. It is these suspensor cells which further undergo cell division to form the globular stage, thereby heart-shaped structure and a mature embryo. However, as a result of the development of suspensor, the apical cell that gets divided moves towards the center or the middle of the embryo sac. Since the repeated division of the basal cell forms a large structure of about 20 to 25 cells, as a result of which the apical cell towards the micropylar end starts moving towards the middle of the embryo sac. Therefore, the large number of basal cells that remains in contact with the apical cell, that is, the large number of suspensor cells which are a part of basal cell, which remains in contact with the apical cell, is called hypophysis. This figure depicts the development of a dicot embryo entire from a single-celled zygote stage to the final matured stage. As it can be seen from the figure, that initially the zygote divides into two, that is apical and basal cell, which further, by developing suspensor cell, develops a four-celled stage, that is by the two times division of the apical and basal cell, which further divides to form a eight-celled stage. Now these division of these four cells occur at the right angle to the initial one. Further division results into the formation of 16th-cell stage, which is the proembryo structure. In this manner, in the 16th cell structure, the two octants get formed. The one that is towards the chalazal end and the other that is towards the micropylar end. Now, the one that is located towards the chalazal end is known as chalazal octant or apical octant. On the contrary, the basal one is known as micropylar octant or basal octant. Thus, division of apical and basal cells forms a four-celled structure which divides to form an octant, that is a eight-celled structure. However, this division is always in the form of right angle of the previous cell. Further, this octant divides to form a 16-cell structure which is known as a proembryo. This proembryo further divides to form various shaped structure which further develops into a mature embryo. Now, this 16-cell structure occurs in the form of two octants, that is, one towards the chalazal end and other towards the micropylar end. As said earlier, these two octants occur at right angle direction to each other. Therefore, the one octant that is located towards the chalazal end is known as chalazal octant or apical octant. And the one that is located towards the micropylar is known as micropylar octant or basal octant. Now various structures like plumule and radicle develops out of these octant. The chalazal octant helps in the development of shoot apex, epicotyle region and plumule. That is the region from where shoot develops. On the contrary, towards the micropylar octant, regions like hypocotyle and radical development occurs. Now, this figure depicts the development of various tissues within the embryo. As it can be seen that towards the micropylar end, that is the peripheral region of the radicle, the root cap develops within the embryo, and that towards the chalazal end, the plumule occurs. This was all about the dicot seed. Now comes the monocot seed. As it can be seen from the figure, the monocotyledons possess only one cotyledon. This is an example of a grass family where the cotyledon is known as scutellum. That is, in monocot plants, the cotyledon is called scutellum. The embryonic axis, this embryonic axis remains attached. In monocots, at one end of its axis, there occurs a structure known as coleoptile. This coleoptile occurs at the end of plumule. There occurs another structure known as coleorosa, which occurs towards the radicle. Thus, coleoptile occurs as a protective covering of plumule. And the second structure, that is coleorhiza, occurs as a protective covering of radicle. Thus, this was all about the embryo development within monocots and dicots. Following embryo development, there occurs and the last stage of the post-fertilization event, that is formation by the maturation of ovule and